Okay, so the three major subdivisions, you've got the plasma membrane, which is going around the outside. It's almost like this is a little biodome, or a biosphere, actually, not just a dome, but a whole sphere going around. The contents that are inside the cell are very different than the contents that are out of the cell. So it's almost like it's its own living organism. In reality, it is the functional unit of life. I mean, this thing is alive on its own, but it depends on this community of other cells that it's in. The plasma membrane gives it its own individual identity in this group of cells. The next is the nucleus, and the nucleus is holding what? What's so important in the nucleus? The DNA. The bulk of the DNA is actually in the nucleus. Does anybody know where the other place you have DNA is at? This is tricky. There's a little bit of DNA inside the mitochondria. So there's only a handful of genes in the mitochondria, less than a dozen of them, or around a dozen of them, that do specific things to make what in the mitochondria? What does the mitochondria make? ATP. So those enzymes that are in the mitochondria are there, or enzymes. Those genes in the mitochondria are there to make enzymes that produce energy. That's their whole purpose. They think this used to be a piece of bacteria, and then your cell swallowed it, preparing to eat it, and went, hey, this is kind of handy. It's making a lot of energy. So it actually became part of the modern cell. So the nucleus has DNA in it, and the nuclear envelope is actually 2 layers thick. That DNA is so important that it double wraps in a double bilayer. So in other words, it's not a phospholipid bilayer like the outside membrane. This is a double phospholipid bilayer. And I'll show you a little bit more detail of that when we get to it. And then the cytoplasm is everything in between. Cyto means cell. Plasma is what this is telling you. So this is actually the cellular plasma, or the liquid that's inside of the cell. When you look at blood and plasma are not the same thing. They're not synonymous. They're not exactly the same. Blood is that whole, everything that's flowing through your blood vessels. Plasma is the watery, liquidy part of blood. Plasma is there with water and electrolytes and some proteins and sugars and things like that. That's plasma. Cytoplasm is the same thing. It's water with some electrolytes and some sugars and fats and things floating around in it, including organelles. So those are the 3 major subdivisions. The first one you want to pay really good attention to is the plasma membrane. Like I said, it's a phospholipid bilayer. So these little things called phospholipids, they're special. They're a lipid, so part of them loves fat and hates water, but they're very unique because these little tiny heads on the top of them are kind of water-loving. So the tails down here are very fatty. That's the lipidy part. But this part up here, the phosphate, actually has a mild charge on it, and that charge loves water. If you remember from your chemistry or biology, if something's charged, it has one of 2 symbols over it. What are the 2 symbols? Plus and a minus. It has a plus and a minus. Water has a slight plus and a slight minus. So anything that has a plus or a minus, or a slight plus and a minus, they love each other. They mix well together. These little phospholipid heads have a slightly charged into them. So they like water that has a slight charge to it. I'll show you more detail in a second. These lipids on the inside have no charge. So are they going to like things that are charged? Like likes like. So things that are alike like each other. If it has no charge, it's going to be really fond of lipids. If it has a charge to it, it's going to be really fond of water. Anybody know the term for that? If you love water, you're hydrophilic. If you hate it, if you hate water, like lipids hate water, they're hydrophobic. They have a fear of water. So this membrane is super important because it prevents things from just going back and forth across the cell wall. If it's hydrophobic, it's hard for the cell to keep it out because it can just slide through the wall. But if it loves water, has any affinity to water, it gets blocked. So what drives me nuts about a lot of textbooks is a lot of textbooks will say, well, water just slides right through this. Will water slide through this lipid layer? No, it hates it. It hates it, repels it. What you actually have to have is some kind of a channel or a pore, a hole, that we'll talk about in just a little bit, that allows water or water-loving substances to get through. There was a study done a long time ago that proved that water doesn't just easily seep through a cell, and a lot of textbooks just aren't up with that yet. All right, so that plasma membrane prevents things from going in and out easily. Of course, the nucleus stores the DNA. It also has some RNA in it. Anybody know what that center part of a nucleus is called, where you have all kinds of activity going on? Nucleolus, right, it has a nucleolus in the middle of it. And we're not going to talk a lot about genetics. I know your book has like 3 or 4 pages on genetics, but we're not going to spend a ton of time on genetics. The organelles that are on the inside, you have to know all of them. If it's on this picture, you have to know it. Wink, wink, star, star, hint, hint. I know the picture's not up here, but these are all listed for you in the next section. All of them. Every single one of them. So get that image. Remember, you have over 200 different types of cells in your body. You have lots of cells. The nice thing about all the cells in your body, except one, all the cells have exactly the same components. Like the cells in your body, they all have nucleus except what cell? Nobody knows this? I'll give you a hint, it flows through your blood. Red blood cells. Red blood cells don't have organelles and they don't have a nucleus. So in other words, when you hear all those forensic analysis and they take a sample of blood and they check it for DNA, are they actually pulling red blood cells out of it? No, they don't care about the red blood cells. Guess what type of cells they're looking for? The other blood cell, the whites. When we do a karyotype, which is taking your chromosomes, we pull a white blood cell out and we do this karyotype. We freeze it and then split it apart and look at your chromosomes. The red blood cell is pretty much worthless. It does one thing. It carries oxygen, or I should just say it carries gases, oxygen and CO2. Okay, so nucleus, what you have to know, of course, the nucleolus, the DNA. Everything that your cell can make comes from DNA, everything. So is a red blood cell good at making new products? Has no DNA. The DNA is like blueprints. I always think of the DNA like my grandmother's recipe book. Did I tell you this analogy already? So my grandmother had this recipe book, and it was as old as she is, probably like 500-600 years old, something like that. But she had it in her kitchen, and it never, ever left her kitchen, because she knew if it went out of the kitchen, there's a chance she'd never see it again. The DNA is exactly the same. The DNA never, ever leaves the nucleus. It always stays in the nucleus. If it leaves the nucleus, it gets shredded. There are little sensors, little enzymes out in the cytoplasm that detect DNA, and they chew it up, because they know that it's flawed if it gets out there. So the DNA holds every recipe for everything you make in your cell. Whether that cell is a neuron or a muscle or whatever, the DNA holds the recipe for it. If something like an X-ray breaks up that piece of DNA, that recipe page in the recipe book, can you make that thing anymore? Nope. When you get a cancer, what's happened is that this DNA has changed. It's mutated. It's been broken so many times that the cell it loses its identity. It doesn't know what it is anymore, and it starts becoming this cell that can become anything. And unfortunately, it usually becomes dangerous and wants to destroy other cells around it, which is a cancer. So the DNA is kind of like a recipe book with all the recipes for anything you want to make in the cell. If you damage that recipe, that cell can't make that anymore. So like a red blood cell that has no DNA in it, it can't make anything. So can it copy itself and replicate itself? Nope. Can it fix itself if it breaks? Nope. It wears out. It has a shelf life. Your red blood cells only live for 120 days, and then they're so old and worthless and they can't fix themselves, so they get shredded. When you look at the nucleus, it's kind of cool. It almost looks like the moon there. You have all these little craters all over the surface, and these are called nuclear pores. If you zoom in on them, they're little tiny holes. Do they let DNA go back and forth? No. But what's the other thing that DNA makes that can get out of the nucleus? It's also 3 letters long. RNA. So RNA can slide through there. If you want to make a copy of Grandma's recipe book, since she wouldn't let it leave the kitchen, she doesn't believe in copy machines, right? So you can't take the book out, copy it, and then bring it back. What'd she have you do if you want a recipe? You have to handwrite it. So when you want a copy of this DNA and you want to be able to use it, you actually take a note card in there and you write on that note card and you copy the recipe. That's called RNA. That one recipe is RNA. Then that RNA leaves, it comes out of the nuclear pore, and it comes out here to the cytoplasm to be made. So out in the cytoplasm, most of these things are actually carried. Originally when we were looking at the cell, we thought, well, this is just like globby looking thing and everything floats around and just haphazardly works. We were so wrong. Inside the cytoplasm, you have so many structures that keeps everything nice and tidy and well organized. You have the cytosol, which is the liquidy fluid. It's almost like being under the ocean for things to swim around in. It's this liquid solution. Cyto means cell. Sol is solution. Full of things like sugar and electrolytes. The only things that really kind of float around in there are things called inclusions, like glucose. What is glucose? It's a fancy word for sugars. It's energy. So you have all these sugar particles floating around. You have different pigments, like if it's a skin cell, you might have something like melanin that's floating around in there that gives that color. You could have steroids like cholesterol that's floating around in there. Everything else is transported by this structure called a cytoskeleton. The cell has a skeleton just like you do. It gives it kind of a shape. It gives it some support. It helps it move things through the cell. If you look really close at these structures, here are some of the cytoskeletal elements. You have a microtubule. This microtubule is like a railway system. So if you want to move something like a mitochondria, if you want to move from one side of the cell over to the other side, there are little figures called dienins that actually grab a hold of the mitochondria and they carry or walk it along these tubules. It's amazing how structured the cell is. This little tiny thing you can't see with your own naked eye has such complex structure. It's amazing. If you ever get a chance, Google Harvard Inner Life of the Cell and watch it. And it shows inside the cell. It's like a 3D animated video of all the things that are happening. You can watch this tubule being built. You can watch those little dienins walking back and forth on the tubules. You can see all the organelles moving all over the place. And if you're lucky, you get the one that has it all labeled, so it tells you this is the mitochondria, this is a lysosome, this is a microtubule, etc. But these are the three major structures, and I think they're listed on here. Yeah, they are. And then we're going to talk about the organelles. So first we're going to talk about the cytoskeletal elements. The three. The microtubules are large tubes. They're like steel pipes. So you have these thick steel pipes. Think of when you go into a factory or any kind of a business and you can see those big steel pipes going through the ceiling. They're carrying water. The water pipes, are they very flexible? No, they're very firm. They're very rigid. That's what these microfilaments are. When you look at them, look how stiff and straight they are. They're these big tubes. They give a lot of firm support. The tail of the sperm is made out of microtubules. So it doesn't actually curl around. It bends and it whips back and forth. My imagination always makes some kind of creation of how I can link this with the real world. Anybody ever been to the top of the Empire State Building? Wow, nobody in here? One person in here. Amazing. I will never go up there because I learned a fun fact for you. At the very top, it will sway 10 feet to one side at any time and then sway back and then another 10 feet to the other side. So you've got a 20-foot span going back and forth. That's saying that if I'm standing here, that thing can sway over this, about this far to one side and then sway that far to the other side while you're standing on it. It's a steel structure. It's this long steel structure that's lots of stories high. When you look at these microtubules, if they're put together really, really long, they can sway back and forth. And that's what the flagella on a sperm does. It sways back and forth with these tubules rubbing against each other. And they look kind of like this. So you can see them all going all the way to the top, and then this tail can go back and forth. Next are the microfilaments, and these are the teeny, teeny, tiny ones. These we're going to talk about a lot this semester. Actually, 2 weeks from now we're going to talk about them extensively. But they look like a chain of pearls. You have a pearl here, you put them together, and then you start twisting two of them. Almost like a pearl necklace. You twist it around. Is it flexible? Yeah. If I pull on it, is it strong? No. If you pull on this, what could happen? Falls apart and breaks. So when we talk about muscles, we're going to talk about these things. You've got lots of these little fibers or these little microfilaments inside of muscles, helping give them contractile strength. But they're not, they're not unbreakable. And we'll talk about that too. And the last one, the intermediate filaments, we'll talk about this in muscle too. So you've got these microfilaments, and then they're covered over with something called an intermediate filament. So that when you contract a muscle, these pull on each other. These intermediate filaments, they're kind of like rope, where you have lots and lots of little strings, but they're all twisted together, and then you twist more of those sets together and more of those sets. So if they're very strong, are they flexible? Can you bend a rope? Yeah, they're very flexible. So they're more flexible than a steel tube, but they're also very strong, stronger than the microfilaments, the little pearl necklace. Places you see it are places that you have a lot of strength in the structures, like hair. If you took hairs and you pull on a hair, it actually takes a lot of strength to break a hair apart, or a lot of pressure, I should say. Or skin, you can pull on your skin and you don't rip it right off because it's held together with something called collagen. That's a bunch of intermediate filaments. So if you look inside the cell and you put a green fluorescent protein on top of those intermediate filaments, you can actually see them running all over the place. They're giving strength and support. They're not perfectly straight like the microtubules. They kind of curve and twist, but they're holding things in place. They're helping support things. Where do you find keratin, by the way? Skin and hair. Yeah, keratin. It's that waterproofing substance on the surface of your skin. It also gives strength to the hair and the nails. Myosin's a fiber inside of muscles. Right. And now these organelles, you have to know these. You have to know them cold. You have to know everything I'm going to tell you about them. that I tell you specifically you have to know. I don't know if I made that clear enough. So the first one, the endoplasmic reticulum. When I was talking about you have the blueprints in the nucleus, I kind of imagine this little biosphere as its own planet or its own city, I guess is a better example. In the very center of the city, you have this library with all the blueprints for everything you need to make, all the recipes for everything you need to make right in there. If you want to make something, you don't want to walk it across town. You want to go ahead and get the copy of your recipe and pull it right outside of the library and go into the kitchen where you're going to make it. So you can think of this like a warehouse or a kitchen. This is called the rough endoplasmic reticulum. RNA always makes one thing. RNA always makes a protein. The structure inside the cell that makes the protein is called a ribosome. And you probably knew that already. But the ribosomes make proteins. They're like little tiny protein maker like workers. They can be free, and you can see a few of them out here in the cytoplasm, or they can be bound to the endoplasmic reticulum. If they're bound to the endoplasmic reticulum, we call that endoplasmic reticulum the rough endoplasmic reticulum because it's studded with all these little ribosomes. Bedazzled, maybe, whatever you want to call it. So the endoplasmic reticulum, the rough ER, that's the factory. The ribosomes are the workers that are attached to it. So the rough ER you have to know is making what? What's being made in the rough ER? Proteins. So the RNA and the ribosomes, their job is to come together. It's like a recipe coming up to the worker, and they take that recipe and they make it. And they always make a protein. As you get a little bit further away from the nucleus, you can see that the ribosomes aren't on this part of the ER, so we call this the smooth ER. Since the rough ER makes proteins with the ribosomes, what do you know that the smooth ER without ribosomes can't make? Can't make proteins. What other biomolecules do you usually associate with proteins? Proteins, blanks, and blanks. Protein, lipids or fats, and carbs, right? So the smooth ER is going to make lipids and carbs where the rough ER makes proteins. These are all factories. You just have this subdivision of the factory where you make the proteins in one area and you make the lipids and the carbs in the other. So when I want to make one of those phospholipids for my plasma membrane, where am I going to make it at? Am I going to make it in the rough or smooth ER? In the smooth ER. If I want to make sugar to store for later, sugar is a form of what? Protein, carb, or fat. It's a carb. So where could I make or modify these sugars at? In the smooth ER, right. So hopefully it's coming together. Another thing the smooth ER does is it helps detoxify things. It helps clean up the environment. So it may take some kind of a toxic substance that you ate, and it'll break it apart and make it a little bit more neutral. So here's just the picture again. There you have the nucleus branching off immediately is the rough ER, and you can see how it's all studded. And then a little bit further out, you have the smooth ER. And it looks kind of jelly-like, like lipids. All right, once you make these products, the endoplasmic reticulum is like a factory, but they make their product well. They just suck at shipping it. So if you make something and you want it shipped, who do you call? FedEx, UPS, USPS, if you're desperate, right? Sorry, if you're a postal worker, don't go postal on me. But you need some kind of a packaging and shipping department. The Golgi is that packaging and shipping department. So you take these brand new made carbs, these brand new made proteins, and you send them over to the Golgi. And the Golgi to me looks like a bunch of pancakes. Here we have one, another one overlapping, another one overlapping. So they're a series. They're not all connected, but they kind of wobble over each other, where it's like my two hands just hovering. What they do is they take these little vesicles, these transport vesicles, from the endoplasmic reticulum, and they absorb them. So they absorb them in the end that's closest to the endoplasmic reticulum, and then they start sorting. They'll take all these, in this example, you can see different colored proteins. So all these different colored proteins, and they start sorting them. They'll start sending them back and forth. They reshape them, they bend them, they tweak them a little bit, and then they put a new packaging label. I mean, they literally put a shipping label on the end of every protein so that that protein has a specific destiny, just like a shipping label goes on a package or an envelope that you send. By the time it gets through to the other end of the Golgi, you can see that all of the proteins are sorted in their own vesicles by where they're being shipped to. Some of them will be shipped out here to the outer membrane of the cell, and they'll be spit out. What's that process of spitting out? Exocytosis. Some of them will get shipped over into another area of the cell like this for use later. Like it may have enzymes for digestion. The cell has to eat, right? So it has to have some kind of a stomach. So this one might have some digestion enzymes in it. We'll talk about that later, which is called a proxy zone. so that later when you eat some bacteria, you use that stomach with those digestive enzymes inside the cell to digest the bacteria. All right, so the process of moving it out of the cell is called exocytosis. A process of moving things into the cell or pulling it inwards is called endocytosis. When a white blood cell eats a bacteria, it doesn't actually break open its membrane. What it does is it wraps its membrane around the bacteria and swallows it, and endocytoses it. If it's a large particle like a whole bacteria, it's phagocytosis. Phago or phage means to eat. So when you hear about a macrophage as a type of cell, what is it? What's its purpose? Macro means big. Phage means to eat. It's a big eater. It eats big things, like it eats whole other cells. It eats cells that don't belong in your body. If you get a disorder called aphagia, what are you having a problem doing? A means without. Swallowing. You can't swallow. You can't eat things. Aphagia, you have something wrong with your esophagus. Maybe it doesn't relax and you can't swallow, or you have a problem somewhere along there where you can't swallow. So when you learn some of these words, it's kind of handy. How many people have taken a medical term? It's a lot of people, more than I expected. All right, pino means to drink. So it's not a big. Phago would be like you eating a cheeseburger. You take big bites of something. Phagio is you slurping that Coke that came along with it. Taking little tiny particles. And then receptor mediated means that it's specific to one particle. Like, for instance, insulin. Your cells, if your cell doesn't have a receptor for insulin, it doesn't know what insulin is. It has no idea. So you have these little receptors across the top, and when the cell is exposed to insulin, it actually sticks to insulin and pulls the insulin inside. And then it'll destroy the insulin. It's a one-time use. When we get to the endocrine system, we'll talk about that in a lot more detail, but it's specific to one thing. Pino just drinks little particles. Phago means eats big particles. And receptor mediated means it's specific to one type of substance. And here's what the Golgi apparatus does. So now you've sorted these things and you're going to ship it to different parts. If you exocytose it, here's its pathway. It comes from the endoplasmic reticulum to the Golgi, it gets sorted, organized, and then gets shipped over here and exocytosed. Whoop, spit out. If this is a pancreatic cell, you can say this is insulin. So it comes from the rough ER, because insulin's a protein, goes to the Golgi, gets packaged, shipped, and when you eat sugar, whoop, it pushes it out into your blood. Exocytoses it into your blood. Goes downstream and finds another cell that is sensitive to insulin, and then insulin gets pulled into that cell. Let's say you endocytose a bacteria. You bring that bacteria in, you bring it along, you introduce it to this little thing called a lysosome. It's like a stomach. The lysosome meets up with that other vesicle that just swallowed bacteria, and what they do is they just merge together. You have digestive enzymes in this one, you have bacteria in this one, and they go whoop, and they just merge together. It's almost like you spitting on your food and digesting it outside of you and then processing it afterwards. And then whatever's left over from the bacteria, it just spits out. It's just like you swallowing and then you poop stuff out later. Same idea. But vesicles are these little things that transport around. So if you bring alcohol and your body tries to isolate it into a vesicle, you introduce it to another vesicle that's carrying enzymes or chemicals that break down alcohol, you merge them together, and then you process the alcohol and make it safe for you, so it's harmless. And then here's the picture of endo and exocytosis. So endo means it's coming into, and again, you don't break the membrane. The membrane just kind of like, it invaginates. It goes from being flat and it kind of curves around and it swallows that thing and just brings it in. And then exocytosis is the reverse. You take a vesicle, you bring it up to the surface. That membrane, these are both membranes that are exactly the same phospholipids. They merge together. This opens up and then just pushes out or ejects out the substance. Here's an example of a white blood cell swallowing a bacteria. So you can see it's hunting the bacteria and then it goes through a process of what? Phago, or receptor-mediated endocytosis. Phagocytosis. It wraps around it, and here you can see it just swallows the thing whole. Here's an example of insulin over here. Insulin binds to a receptor, and it slowly starts wrapping itself into a vesicle full of that insulin. And then it brings it inside and chews up the insulin. And here's an electron microscope picture of what happens with exocytosis. It's kind of cool because you've got this high concentration of product in the vesicle, and as soon as you open it to an area that's low concentration, so what happens? If you've been in lab, you know what happens. High to low concentration, what's that process? Diffusion. It diffuses out, quickly diffuses out because it's high concentration in here. It almost looks explosive, like it's blasting out. I think I'm just amazed with the molecular biology of one cell. If you ever take a molecular biology class, their textbook is twice as thick as yours, and it's talking about one thing, a cell. That's it, the whole time. When you look at this vesicle as it's coming up to the surface, it has these little twist tie structures called snares. They meet up with the outside membrane and they twist together. They just naturally start twisting together until it peels apart that vesicle, and then everything inside is ejected out for exocytosis. All right, some of those vesicles have special structures in them. One's called a peroxisome and one's called a lysosome. The lysosomes are kind of like the stomach. Lysosomes contain enzymes that are hydrolytic. So a hydrolytic enzyme, what's hydro mean? Water. Lytic means it's doing what with water? Lysis or lytic means to break. So it's using water to break apart things. That bacteria, it's almost like you're taking water and enzymes, exposing it all over the bacteria, and the water just cuts right through the bacteria. So it's the enzyme and the water that work together. And then the final product of a lysosome is usually some kind of a toxic product, like a free radical. If you've heard of free radicals or reactive oxygen species, those are the things you take antioxidants to get rid of because they're so bad for you. The antioxidant neutralizes that free radical. Lysosomes, when they're overactive, they make lots and lots of free radicals that can damage the cell. So when you drink lots of alcohol and it goes into your body and your body's trying to break all this stuff down, you're making all these free radicals that are actually toxic to the cell. Same thing when you eat overcooked meat. So there are a lot of problems that happen. Peroxisomes use something called hydrogen peroxide, which is H2O2, to break things apart. So it uses hydrogen peroxide, which is actually a free radical. It destroys whatever you're trying to break down, which is primarily lipids and alcohol. So peroxisomes really like to attack lipids and alcohol. And then its byproduct is water and oxygen. Are either of those really toxic? No. I remember when I was taking chemistry, there was this thing about somebody that called a school and complained because all the drink fountains had dihydrogen monoxide in them and kids were drinking it and it could make them really sick. And some people were panicking because the dihydrogen monoxide was getting in their kids were drinking it. You know what dihydrogen monoxide is? 2 hydrogens and 1 water, or 1 oxygen. It's water. So they were right. They just weren't exactly accurate in it being toxic. Okay, so that's a big difference. Lysosomes start with something good and end with something bad. Peroxisomes start with something bad, hydrogen peroxide, which will, if you put it on your hair, it strips the color out of the hair, it denatures the hair and changes its shape and everything. It's bad, it's toxic, but your cells make this stuff. And when it uses it, it actually makes it into something good, water and oxygen. They have exactly the opposite effects. Exactly the opposite process. And then the mitochondria, and of course the mitochondria is the power source. And if you open the book, it spends, I think, 2 whole pages talking about the mitochondria, but I'm going to make it really simple for you. What's it make? ATP, which is the energy source of the cell. When you see ATP, you always know that it's the energy form the cell uses. It's a high energy storage unit. A mitochondria will take one little piece of glucose, split it apart, and then the mitochondria pulls it inside and makes it into $36 worth of ATP, 36 pieces of ATP. So some books say 34, some say 38, 36 is what we're sticking with, right down the middle. So like I said, it'll split this sugar out here, and then it pulls it in, and it weaves it back and forth across this membrane. You can see all that membrane inside. As it's weaving it back and forth, it's using oxygen, and it's using oxygen to split these little tiny electrons off, and finally on the other side, out comes carbon dioxide and ATP. So when you're making lots of energy, you're also making lots of what? Carbon dioxide. Think about it. When you exercise really hard and you're burning lots of energy, you're making lots of energy and you're burning it up really quick, what are you making lots of at the same time? Carbon dioxide, which makes you do what more? Breathe more, panting, trying to get it out. Because if you don't get it out, your body becomes really acidic. We talked about that the first day. And we'll talk about it over and over again. And then plus, because you're burning lots of oxygen to make the ATP, why else are you breathing heavy? To get more oxygen in. So this process is actually called the citric acid cycle, and this chain is called the electron transport chain because it's transporting electrons back and forth to make this ATP. It's almost like a nuclear power plant inside of your cell. It has all these really delicate processes that have to be perfect for everything to work just perfectly. So what would happen if you didn't have oxygen? What could you not make efficiently? ATP. You can't make ATP efficiently. I'm saying efficiently. Can you make ATP without oxygen? Yes. Can you make it with the mitochondria? No. So you have to have some other process to make ATP. Any of these raw materials can make ATP, but the best one, the ideal one for your body is glucose. When you store glucose, it's called glycogen. It's the storage form of glucose. So when your body, let's say you sleep all night, when you eat a meal, you've got it in your stomach for about 4 hours, your intestine for another hour or so. So as it's moving down through your GI tract and you're sleeping for 8 hours, you only have energy from a meal for about 4 to 5 hours. So after that 4 or 5 hours, you need to start pulling out these storage energy forms. You need to start burning fat, you need to start burning protein, you need to start releasing sugar into your blood. Anybody know where the main storage site for sugar in your body is? What organ? It's the liver. When you eat sugar, your body will put it in the blood and then it starts storing a lot of it in the liver as glycogen. It's a storage form. Later, when your blood's running low on sugar and you're feeling really tired and drained, your liver will start dumping that sugar back into your blood. It's a loaner site. If your liver starts running out of sugars, then you start breaking down fats. So then you start pulling it from what part of your body? The adipose tissue, which is in your butt cheeks and love handles, right? Primarily. So it starts breaking down fats. And if you start breaking down too many fats, then your body will start breaking down proteins. Where do you store protein in your body? Muscles. So when somebody's starving, let's say you get trapped out in the woods and you have nothing to eat. You start breaking down your sugar for about 4 or 5 hours. Then you start breaking down your fat. But as you start breaking down fat, your body is going, man, this fat's really good for energy, but it's releasing lots of toxic waste products at the same time, which we'll talk about later. So it starts breaking down protein. It starts eating your muscles. Well, if you haven't eaten for a few days, your fat stores are going to start dwindling down, and your muscle stores are going to start dwindling down to the point where you stop eating skeletal muscle and you start eating what kind of muscle? Cardiac muscle. And that's when people die from starvation. You don't die because you just starved. You die because your heart is actually being eaten right out of your own body, and it can't work anymore, and you die of a heart attack. There's a fun fact for you. So the mitochondria can use all these different forms for energy. But what does the mitochondria have to have? Otherwise, none of those forms work. Did you catch that? It has to have... What up here did I say you have to have? Oxygen. You have to have oxygen for the mitochondria to work. Right. So what happens is you break that sugar down first through a process called glycolysis. Glyco is referring to what? Sugar. Lysis means you're doing what with it? Breaking it. So you take this sugar particle and you split it in half. And right now it can actually be made into energy without oxygen, but if you give it to the mitochondria and say, here, eat this, it'll spit out 36 bucks worth of ATP on the other side, like this. So it takes that piece of sugar, break, and then spit it out over here. So about 36 pieces of ATP. If you have oxygen. If you don't have oxygen, this glycolysis works, but you need to do something with those 2 pieces of sugar. So what do? You turn it into 2 pieces of ATP and something called lactic acid. Ever heard of it? Lactic acid is an acid. It's going to start eating your proteins. It's going to start causing problems with your proteins. It's bad stuff. So if there's no oxygen present, you can't use this process called oxidative phosphorylation in the mitochondria. You can only use glycolysis, which makes 2 ATP for 1 piece of sugar, plus lactic acid. So when you start exercising really hard and the oxygen is not getting to your muscle efficiently, can your muscle still make energy? Yes, but what can it not make? Or what can it not use to make the energy without oxygen? It can't use mitochondria, so it uses this glycolysis, and it's only making 2 pieces of ATP, which isn't enough energy to keep the muscle going. That's when your muscles start failing. And it also keeps making what other bad chemical? Lactic acid. So if you work out really hard and you know you should stop because you can feel the burn in your muscle and it's burning really bad and you're just pushing and pushing and pushing, and you have all this lactic acid building up, how do you feel the next day? Sore, because that lactic acid is going to eat the proteins inside your muscle, and it makes a lot of problems. We'll talk about that more when we get to muscles in 2 weeks. So there are two processes you have to know. If you have oxygen, you can use the mitochondria and do something called oxidative phosphorylation. Oxygen, oxidative. Those words go together. How many ATP do you make in that form? 36. If you don't have oxygen, can you use the mitochondria? No. But what's the other G word process you can use? Glycolysis, right. Glycolysis. How many ATP can you make? 2. But what's the other bad byproduct? Lactic acid. By the way, that process is called fermentation. If you do it in yeast, it actually is great because it makes good, delicious beer. It makes really delicious bread. But in us, it makes lactic acid, and it's really bad. Okay. When you see this word aerobic pathway, aerobic means oxygen is present. You use the mitochondria. Anaerobic will be the same as saying glycolysis. So there are a couple terms you need to remember. That glycolysis, anaerobic, without oxygen, makes 2 ATP and makes lactic acid. When you're talking about an aerobic situation, you use the mitochondria. It uses a process called oxidative phosphorylation. It uses oxygen. It makes about how many ATP? About 36 ATP and some CO2. Okay. So the cell can make more ATP aerobically than anaerobically. It's kind of the big picture here. And then the last structures are the vaults. Vaults are relatively new. We've only known about them for a short time. They look like symbols to me. So here's an open vault. It's like 2 symbols that you see in the orchestra. And what happens is that if you get some kind of a toxin that your cell doesn't know how to deal with, it's not like alcohol that you can process. This is a toxin your cell doesn't know what to do with. What we believe is happening is that that toxin will float in here, and that vault will see it and go and clap together. When it claps together, it kind of bends into this barrel shape over here and traps that toxin. The reason we think this is what's happening is that people that take chemotherapeutic agents for cancer, we find that they better deal with chemotherapy if they have a lot of these vaults in their body. So if the cells in their body make a lot of vaults, they can deal with chemotherapy a lot better. If their cell doesn't make very many vaults at all, they get really, really sick, like the extreme end of sick. So we think that this is almost like when a bacteria becomes resistant to a drug. There are certain processes on the inside of the bacteria that trap that drug and pump it back out. We think the vault's kind of the same idea. When you get exposed to this bad toxin, it's your way of becoming resistant to that toxin. And that's the only significance to a vault. So if I asked you a question about these organelles, and I said, where's the energy produced for cell life? Is it number 1, the endoplasmic reticulum? What's made in the endoplasmic reticulum? Proteins, carbs, and fats. Which part of the endoplasmic reticulum makes the carbs? Smooth. Which part makes the proteins? The rough. Which part makes the fats? Smooth again. How about the Golgi complex? What's its purpose? It's the packaging and shipping department, right? How about the lysosome? What is it like? It's the stomach. It uses water and enzymes to digest things. How about a peroxisome? What's it do? It uses what? Hydrogen peroxide, and then it puts out what? Hydrogen peroxide, it detoxifies things like alcohols, it breaks apart lipids, and it makes water and oxygen. And then the last one, mitochondria, that's where you make how many ATP per 1 piece of sugar? 36, if you have what present? Oxygen. Did I beat that to death yet? Okay, next structures, the plasma membrane. The plasma membrane, as we go through this, there are 3 major components you need to get familiar with. And the first is that it has this lipid bilayer. Does anybody remember the name of that lipid molecule that's in the bilayer? The primary lipid molecule. Phospholipids. Yeah. We'll actually talk just briefly about another lipid that's in there. It's called cholesterol. But the primary lipid that's in here is the phospholipid. What was the unique feature about the phospholipid? These little heads love water and the tails love fat. Yeah. And then the membrane proteins, these are the things that go through and help things that are water-loving get through. So here you can see a pore or a channel, so something that loves water can't get through the lipid membrane, but it can slide right down that tube. Other proteins are there as like receptors. almost like a light switch on the outside of the cell. So if you want to do something inside the cell, you just walk over to the switch and flip it. It's a receptor. Other ones might be to tie one cell to the next. So grabbing a hold or supporting and grabbing. I said grabbing twice. A little redundant. Just a little repetitive, redundant. And then other structures might hold sugars in place, and I'll talk about that in a second. And the carbohydrates, by the way, are the sugars. The carbohydrates always sit on the outside, and they look like little branches. What's kind of funny is that when you look at an actual cell, all those branches are so close together, they look like cotton candy across the surface of the cell. And the important or significant thing about the carbohydrates is that they are actually a fingerprint for the cell. They help the cell with recognition. So when one human cell comes over to the other human cell and they meet each other, they go, oh, hey, I know you, you're the same person's cell. If your cell comes up to my cell, they look different. The fingerprint on yours look different than mine, and what will they do to your cell if it's in my body? Kill it. They'll reject it. The bad news is this fingerprint is also identification for things like virus, HIV, HPV. They come along and these little tiny viruses look at that membrane, that fingerprint, and they grab hold of it and they say, oh, perfect, a human cell, and they pull itself in. That's why HIV is specific to humans, where SIV, simian immunovirus, or immunodeficiency virus, is for what animal? Simians. Monkeys, right? So we don't get the simian version, we get the human version. It came from the simian version, but adapted. And then there's FIV, which is the feline, which is what? A cat. All the fingerprints on top of these living cells are different. So HIV only recognizes human, where FIV is for felines and SIV is for monkeys. So carbohydrates are there for identification, if I didn't say that 3 times already. Recognition, identification. So let's talk a little bit more about those lipids. And you already know that the primary part of the lipid is the phospholipid. It has a polar head. Anytime you see polar, you want to think charge. So does it like water or dislike water? Likes. Water is polar. It has a charge on it. Polar things like polar. It's almost like magnets. If you take a north and a south end of a magnet, and you take one south end and put it next to another south end, what do you know about them? They push off each other. If I put a negative end by a negative end, what are they going to do? Push off. But what if I let the two magnets sit close to each other? What do? They spin around and they love each other now, right? That charge pulls them together. What if I take a magnet and put it next to a piece of plastic? Does it like the plastic? No, because the plastic doesn't have a charge. There's no charge to it. It doesn't want anything to do with the plastic. I can put it next to the plastic and it doesn't repel the plastic. I can flip it around, it doesn't care. It has no affinity or any repulsion to it. It just doesn't care. The lipids are like plastics. They prevent anything from getting through that's non-plastic. There's a close-up of one phospholipid. Here you have that polar head with the charge on the end of it. Here you have the non-polar tails. This charged end likes water, the tails like fat. So when you put them in water, all the heads will line up right next to the water, and they'll actually push all the water molecules out of the lipid area. So they'll push some of the particles out this way, they'll push some of them out this way, but then it forms this wall. Well, the bad thing about the wall is it has this lipid exposed over here, so what do you think it's going to do? If you put them in together, they will find their way. It's almost like taking water and oil and putting them in a container and shaking them up. At first they look like they're mixed, but when you sit them down, what do? They separate. So this thing will keep rearranging itself until you have this continuous ball all the way around. So there's no break. This wall is still connected all the way around here. There's no end to it going this way or around this way or any way that you look at it. It's a continuous ball. So you have this intracellular area and you have the extracellular area that are separated completely by this lipid membrane. Right. So this is not in your notes, but if you go back and you listen to the lecture again, or if you look at your notes and you wrote down what I told you, here are the main points you want to know about the lipid bilayer. It forms the basic structure of the membrane. It forms the outer layer. It separates the inside from the outside. What's so special about the core of that layer? It loves, where the tails are at is the core, they love fats, which prevents what from getting through? Anything that loves water, which is anything that's hydrophilic can't get through. And then because they're lipid, they actually allow some fluidity. They allow it to move just a little bit. The proteins, I already told you, they form channels to let what kind of things go through? Things that love water. In fact, what they found in that study I was talking about earlier is that there's a structure called an aquaporin that specifically allows water to get through. If you have a cell that has no aquaporins, can water get in? Nope. They did this study with frog cells. Frog cells are in water, so if they had too many of these aquaporins, the water would rush in and make them go whoop, pop. So frog cells actually have no aquaporin, which means water can or can't get in. Can't get in. They're waterproof. Frog cell eggs are actually waterproof. Right. They can be carriers, so they can move things back and forth, not necessarily just water, but they can transport. We're going to talk about this a lot more in detail. They can be receptors, like I mentioned before, like a light switch on the outside of the cell. They can help with recognition, and they can help hold cells together. What's the main purpose of carbohydrates? The recognition. When you look at these carbs out here, they sit further up than the proteins. They're the first thing other things see or recognize. Some of the cells, the proteins in the cell can help recognition, but the carbohydrate, the primary purpose of it is cell recognition. Where was I at? There we go. So if I ask you a question like this, which one of the following is not a characteristic of the lipid bilayer? Look at your neighbor and tell them why you're right. Wow, a lot of people that don't feel like they're right. So tell them why number 1 is right or wrong, or if it's true or false. How about number 2, true or false? How about number 3, true or false? Number 4, true or false? How about number 5, true or false? Right. So number 1, basic structure of the membrane is made out of lipids. That one's true. So you can cross that because you're looking for something that's false. You're looking for something that's not a characteristic. Get rid of number 1. How about number 2, hydrophobic interior serves as a barrier to prevent water-soluble substances from going through. That's also true. Get rid of that one. How about number 3? Is it fluid? Is the lipid fluid? Yes, it is. Remember, there's kind of that fluidity, a little bit of mobility to it. So that one's true. How about number 4? Water-soluble or hydrophilic substances can cross through the lipid bilayer. That's false. But don't stop there. Make sure that the last one's true. The hydrophilic heads of the phospholipids face the intracellular fluid and the extracellular fluid. That's true. So number 4 is our correct answer. This is the only thing that's not true. Okay, membrane transport, brand new concept. When you're moving things back and forth, and we're actually going to test this in lab a little bit, you can describe the membrane as being permeable, impermeable, or selectively permeable. Permeable means anything can flow through. Impermeable means nothing can flow through, and selectively means that some things can, some things can't. So in this membrane, you can look at the distance or the size of the holes in that membrane. You can see that water can slide through, but whatever this thing, like maybe sugar, can't. Would you say that that's permeable, impermeable, or selectively? It's selective because it lets some things get through, but not everything. Some of the biggest 3 factors you want to pay attention to when you're asked about permeability are the size of the particle. So the first thing you want to ask, is this a big particle? If it's a big particle, do you think it's going to easily move through a membrane? No, it's like a big guy that's like 400 or 500 pounds trying to squeeze through a little tiny doorway. It's hard to do. Number 2. Are you 400 or 500 pounds? Yeah. Oh man. Now I have to be more careful what I say. Number 2. Lipid solubility. Solubility is saying does it have an affinity or does it like what that membrane is made out of? So, is a water-loving particle soluble in a fat, a lipid membrane? No. So, if I say that this thing is very low solubility, it must love what? So, if it's very low solubility in a fatty membrane, it must love low solubility. It must love water. It's not very soluble. When you see the word soluble, it means can I mix it and it dissolves inside of it. If I mix a fat with a fat, will the 2 fats dissolve together? Yes, because they're highly soluble. If I mix a water and a fat, do they dissolve together easily? No, because they're not very soluble. So if I tell you that a drug If I tell you this drug, like morphine is lipid soluble. Will it go through a lipid membrane? Yes. If I tell you penicillin is not, penicillin is water loving, will it go through a cell membrane easily? No. How about insulin? Insulin's made out of a protein that loves water. Can it move through or into the cell easily? No. So when I tell you something's lipid soluble, you know it's going to get right in. So if I tell you a drug is lipid soluble, you know it's going to move right into the cell. If I tell you a substance, no matter what it is, like estrogen. Estrogen is lipid soluble. Can it get into all of the cells of your body? Absolutely, because every cell is made out of a lipid membrane. Number 3, the charge. Charge means it has a positive or negative charge on it, or it's slightly polar is another word we refer to it as, or polar. So remember, water has a charge. It's polar. Will water like other things that are polar? Yes. Will the fat like things that are polar? No. Fat is non-polar. Fat's kind of like plastic. It repels things that are electric or have charge or whatever. Movement of particle across membrane is produced by some type of force. 2 major categories, passive and active. Passive means that you don't require any energy. Only the energy of that particle does it. So once a particle starts moving across the room, it stays moving by its own energy. For me to change that, I have to put my energy into that particle. That is active. If I put 300 of you in this room and there's nobody in the hallway, where do you want to go? Out. Do I have to force you out? Nope. You passively move by your own energy out of the room. So you are passively moving. If I don't want you to leave and you get out in the hall, and I have the guy back there that was laughing at me earlier, and I tell him I want you back in the room, is he going to have to put energy into putting you back in here? Yeah, he's going to have to force you back in here. So now we have our door bouncer. So active means that something outside of that particle had to put energy into moving it. So what would this picture be? Would this be a passive movement of the boulder or an active movement? It's active. You're pushing the boulder where it doesn't want to be. If I let go of it, what happens? It passively rolls downhill by its own movement. So a lot of times you'll hear the word concentration gradient or down the gradient. When you go up a hill, you're saying you're going up a gradient. Sometimes in some states you can actually see they'll have a picture of the hill and they'll tell you a number under it that tells you the degrees gradient of the hill or the grade of the hill. So what would move faster? What do you think would move along this gradient faster? If I had a small gradient with a little stack here and a little tiny slope, or if I had a huge gradient or a high gradient with a way up here going way down here? The small slope or the large slope? The large slope has a higher or faster gradient, and the low one has a slower gradient. So when we talk about things moving, we like to describe them as moving along the concentration gradient. If it moves along the concentration gradient, it's diffusion and it's passive. So passive and diffusion. If it moves against its gradient, it's always going to be active. Nothing wants to go against this gradient. Even the universe, everything, particles, if I spread perfume up here, it moves from where it's high concentration until it gets all the way to the back of the room where it's low concentration. Our universe is expanding because it feels crowded, and so it's pushing its way out. It's diffusing. It's moving with its concentration gradient. If I had a bunch of particles in water, if I put food coloring, green food coloring in this water, it would start diffusing or moving down its concentration gradient until it all spread out evenly. If I put electricity moves on its gradient, I'll show you in a second. If I put a bunch of negative charges over here and a bunch of positive charges over here, where's the negative want to move? It's crowded here, so it wants to move over here where it's not so crowded. The positive wants to go where? To the opposite side where it's not so positive. So diffusion is the movement of particles down their concentration gradient. Until they hit equilibrium. Over here is an example of equilibrium. This thing's moving down its concentration gradient until it gets to a point where it's even on both sides. You can see it's crowded here, nothing here, so it spreads out. But once it's spread across the entire area, it's at equilibrium. This particle may go over to this side, but at the same time that particle will go to the other side. So as one moves to one direction, the other one will swap places. We call that steady state. So in this situation, where do the little balls want to go? Do they want to go from B to A or A to B? They want to go to A to B until they hit equilibrium. You have to know this guy's laws. All semester we're going to talk about them over and over and over and over and over again. So Fick's laws of diffusion. So Fick did these experiments because he wanted to see what affects how fast things move. What affects how fast things diffuse. The word rate means speed. So he wanted to know what changes the speed of diffusion. And these are some of the basic rules he found. He found that the magnitude of the concentration gradient does it. What you want to know is specific. If I raise the concentration or I raise the magnitude of the gradient, what happens to the speed? It goes faster. If I put a drug in somebody's blood and I put a lot of it in there, is it going to move slow or fast? It moves fast. Next is the permeability of the substance. So you can think that solubility again if you want. But the permeability or solubility is another way to look at it. How easily can you get through the wall? So if I take this wall and I only have 2 little doors, and there are 300 of you in this room, and I say, now we can leave, and I only open that door, are you going to easily move through fast? No, you're going to slowly go through. But what happens if I open both doors and I make it extremely permeable? You can leave twice as fast, right? So your rate of diffusion went up. The more permeable, the faster the rate of diffusion. Next is the surface of the membrane. So how big the membrane is. This is going to be super important when we talk about things like emphysema. What lung is... Well, lung. Good job, dummy. What organ is broken in emphysema? The lung. So your lung, if I took your lungs right now and I ran a steamroller over them over and over and over again until they were 1 cell thick, you would fill a tennis court. So about half of this room is how flat your lungs would be. Somebody with emphysema, if we did the same thing with them, their surface area of their lungs would be maybe half of a tennis court. What's going to happen to the rate of diffusion of oxygen in them? It goes down. They have smaller surface area, so the diffusion goes down. Next, the molecular weight. You can think of the size of the particle or the heaviness. When you're talking about molecular weight, particles are interesting because you can have 2 particles that are actually the same size, but they weigh differently. Think of a bowling ball and a soccer ball. They're like the same size, right? But is there a difference? Which one's going to move faster? The soccer ball. That's why you don't see them kicking bowling balls around a soccer field. So the soccer ball, lighter weight. Its molecular weight is smaller, so it moves faster. This bowling ball is so heavy, so thick, and even if it were bigger, it's the same problem. It's going to be harder to get it moving. So what would speed up the rate of diffusion? Bigger molecular weight or bigger size, or smaller molecular weight or smaller size? Smaller is faster diffusion. And then the last one is the distance. So think of that plasma membrane. Here you have the outer plasma membrane that's a bilayer. If I have something like oxygen moving through a bilayer versus something that's moving through the nuclear envelope, which is a double bilayer, which is take this and double it, what's oxygen you get faster through? The nucleus or the plasma membrane? The plasma membrane, because it has a shorter distance to go. I've always wanted to do this experiment. Make a Jell-O wall, right? So you make a Jell-O wall that's a foot thick, and then you make one that's like 6 feet thick. And then we race through the wall. Who's going to get through faster? The one that has a short 1 foot distance or the one that's doing the 6 feet? The one that's 1 foot. You blast right through that Jell-O wall, piece of cake. I want to do the 6 foot because I want to run at it and just go boom and stick there. I just want to see what that's like. The sucky thing is I'm sure that I wouldn't be able to breathe in there. That's the cost for fun, right? So those are fixed rules and you have to know them. Because as we talk about, like I just gave an example, the lungs, as we talk about the kidney. So if the kidney is more permeable, are you going to make more or less urine? If I say that the wall of the kidney is more permeable, are you going to make more or less urine? More, because things are going to pass through it faster, right? Let's see, if I talk about the thickness, if I'm talking about blood vessels, if I'm talking about blood vessels that are building up plaques, what's happening to the thickness of the wall? It's getting thicker and thicker. Is it going to be easy to diffuse oxygen out of your blood and into your tissues? No. So all of these things apply down the road. Just have to keep them in mind. And then ions. When you see ion, you can think electrolyte, you can think it's polar, you can think it's positive and negative. All of these terms are pretty synonymous. But ions means there's a charge on something. So I'm going to use sodium. I'm going to say sodium has a positive, I'm going to say chloride has a negative. They actually do. So let's say I have a bunch of these positive sodiums over here, not so many positive sodiums on this side. Where's sodium want to go? It wants to go down its concentration gradient. It wants to go from where there's a lot of sodium to where there's very little. It doesn't matter if I said sodium or not. If I say there's a lot of positive on this side of the wall and there's not barely any positive on this side, where's the positive want to go? It goes down. So when you're talking about things moving on their concentration gradient, you could talk about chemicals moving, but you could also talk about electricity moving or ions moving at the same time. So I like to simplify things. I already know the law of diffusion or how diffusion works. A lot of textbooks will go through and they re-explain ions and how ions move, and they refer to it as the electrical movement instead of just saying, you know what, it's diffusion but with electricity. That's all it is. It's diffusion with electricity. Right? Diffusion of water is osmosis. So if it's diffusion of any other chemical, we call it diffusion. If it's diffusion of water, we specifically say it's osmosis. Water always wants to move down its concentration gradient. If I have just 1 little liter of substance over here and it has 100% water in it, but over here I have 10 liters of substance and it has only 10% water in it, where does the water want to go? Does it want to go to the 100% side, or does it want to go to the 10% side? 10% side. It doesn't matter how much other substance is in this 10 liters of substance. Water moves from where water's cramped to where water has more space, where there's very little water at. It always moves like that. Water will defy everything you think about it to try and move where there's less concentration of water, and it's called osmosis. I always think about my house, and I should draw an analogy of the house sometime, but when you think of your house, what percent water should be in your basement? I'm hoping 0. And then it used to rain in Iowa, and one weekend I came home and it was raining, and it was outside. I saw standing water in my backyard. There was 100% saturation of water in the ground outside, hopefully 0% in my basement. Where does the water want to go? To my basement. So what is that doing? Is that an active or a passive movement? It's a passive movement, and it's called what? Osmosis, diffusion of water. It wants to seep into my basement. Luckily, down in my basement, I have a special device called a pump. And that pump requires what to work? Energy. And that pump turns on, and the pump will take that 1% water that leaked into my basement and put it where? Outside where it's 100% water. It tries pushing it where it doesn't want to be. I put energy putting that water back where it didn't want to be. So there you have passive movement coming into my basement, active movement pushing it back out of the basement. That's osmosis, and then pumping of water is the other side of it. So if I have these two structures here, this is 100% pure water, this is what, 90% water. Where's the water want to go if I connect these two? It wants to go over here to where it's only 90% water, until it hits what? Equilibrium. Will it ever hit equilibrium with this side if this solute doesn't move? No, it never will. It'll just keep pushing over, but it'll never hit equilibrium. What if I make this a membrane permeable to that solute and to water? What will happen? The solute will go over here, and the water will go over here until they're both at equilibrium, like this. So if I have this situation where the blue particles are water, the purple ones are the solute. By the way, what's water? It's the universal solvent. So right now I have a solution. Water is a solvent, and these are the what's? Solutes. So I have this membrane, and things can move back and forth. You can see that the solute can move over here, so it'll move until it's at equilibrium. 3 over here, 3 over here. Water will move until it's at equilibrium, where it's the same concentration here as here. But what if I put a semi-permeable membrane that doesn't let the particle move? So if the particle can't move, it bounces off that membrane and stays over here. Where's water want to go? You can obviously see there are fewer water particles on this side just because there are more of the solute. It's the same volume, but the concentration is different. Don't mistake volume and concentration. They are extremely different terms. I can have 1 liter, like I said before, of pure water, and I can have 1 liter of only 10% water. Where's the water want to go? It wants to go to the 10% side. It will push over into the 10% side and actually add, like this. It'll keep moving from where it's high concentration to low until you actually see the water raising and possibly even spilling over the other side of that container. This movement's called osmotic pressure. Osmotic pressure is water pushing across to where it's a lower concentration. It'll keep pushing up until it stops because of a force called hydrostatic pressure. Hydrostatic means water stopping. So it's hydrostatic pressure. You've got osmotic pressure pushing one way, hydrostatic pushing the other way. When those two pressures equal each other, they stop moving. These are two terms we're going to come back to when we start talking about blood in the kidney. You have to know these three terms. Iso, hyper, and hypo. Iso means same, hyper means more, hypo means less. When you're comparing or describing concentrations, you always have to have two places. Are you tall? Are you short? Are you rich? Are you poor? Are you skinny? Are you fat? How do you know what you are? Because you compare yourself to someone else. When you're talking about a solution, you can't describe it as iso, the same, unless you have something to compare it to. So all the time, every time we compare anything this semester in this class, and most of the time in your medical career, when you're comparing a solution in the body, what's the most important solution in the body you can compare to? The only one that you can affect as a medical professional. The only one you can pull out or extract as a medical professional. The only one you can add drugs to as a medical professional. The blood. It's always your reference point. You get sick, you go into the doctor, and they're like, I don't know what's wrong with you, let's take some blood. They want to know what's going on in your environment, so they take this standard thing that they compare everything to. They compare you to everybody else on the plant's blood. So the solution you're always comparing to is the blood. So we're going to use red blood cells as our reference point. If I tell you, I pull out a red blood cell, red blood cells are 0.9% saline, so just for ease of math, we're going to say 1% saline and 99% water. If I take this one cell and I drop it in this glass that's 100% water, that water, quote-unquote, solution that's around it is called what? Hyper, hypo, or iso. Is it the same as a solution in the cell? Nope. Does it have more solute or less solute than the cell? What's the solute inside the cell that we just said? The solute is the sodium. So is the solution outside in the cup that's 100% water, is it higher in sodium or lower in sodium? What's the word for lower? Hypo. So the solution that's 100% water is the most hypotonic solution you can get. So if you want to use 100% water as the most hypotonic, always use that as your reference point. The closer it gets to pure water, the more hypotonic it is. So if I drop a cell, a red blood cell, in a hypotonic solution, and it's 99% water inside the cell and 100% water outside the cell, where's the water want to go? 99% inside, 100% outside. It goes inside the cell. What's going to happen to that cell? It expands, and then eventually it can lyse. What does lyse mean? Break. So is it a good idea if you've been exercising all day, sweating, losing electrolytes, losing water, to start drinking Evian, pure water? Nope. Is it a good idea that if you're dehydrated and you're overheated to go to the hospital and stick a 100% pure water IV bag into your arm? No. If you stick that 100% water bag into your arm, what's going to happen to all your red blood cells? They expand and then they start popping all over the place. I like to think they make that sound. Man, it's a good thing you're not in my head, because I'm telling you, it's super animated. But they start blowing up. It's not a good idea to give a hypotonic solution to somebody, pure water hypotonic solution to somebody that's dehydrated. You can sometimes give a slightly hypotonic, just really mild hypotonic solution to them to help them a little bit, but man, you really have to monitor to make sure you're not destroying their red blood cells. Hypertonic is saying that the solution outside has what? More or less solute in it? More. Hypertonic is referring to the solute. So instead of having 1% salt, it's anything above 1%. Maybe 10% salt. Man, could you imagine if your blood were 10% salt? How would you start feeling all the time? What happens when you eat a bag of pretzels and you bump it up to your blood goes up to 1.5% salt? You feel really thirsty and dehydrated. You can't handle that. Because guess what's happening to all of your cells? They're shriveling up. If you put a red blood cell in a hypertonic solution, that hypertonic solution now has more solute in it, but it has less solvent. So let's say that this red blood cell that was 99% water, out here you've got 10% salt, so what percent water is it? 90. 99% water in the cell, 90% outside the cell. Where's the water want to go? Outside the cell. What's going to happen to the cell size? Shrivels up. It wrinkles like a raisin. When you put a cell in a hypertonic solution, it shrivels like a raisin. If you cure meat, if you take a slab of meat and you pour salt and sugar over it, that salt and sugar has how much water in it? None. What's it going to do to all those cells that are 99% water? It's going to pull their water out. A bacteria cell sitting on the surface of that's going to have what happen to it? Bacteria cells are still about 99% water, so what's going to happen to that water? It gets pulled out into the salt. What's going to happen to the bacteria? Shrivels up like a raisin and dies. Why do you cure meat? To kill bacteria. It's not to make the meat taste salty or sweet or whatever. It's to suck the water out of the bacteria and kill the bacteria to cure it, quote unquote. Oh, I forgot ISO. ISO means the same. So in other words, ISO means if you put the solution, if you put an isotonic saline solution in somebody, it's 0.9% saline, which is exactly the same as their blood. What's going to happen to a cell when you put it in an environment that's the same as a cell? It's at equilibrium, so it does nothing. It's happy there. A cell is surrounded by a hypotonic solution. What will happen to the cell? It swells. I also look at these words, so if you don't understand the concept completely yet, look at hypotonic. When you put a cell in a hypotonic, I imagine that O going and expanding and popping. If you put a red blood cell in a hypotonic solution, it's going to swell up and pop. If you put a cell in a hypertonic solution, what word or what thing can you imagine with the R? A raisin, right? So if you put a cell in a hypertonic solution, you make it look like a raisin. It shrivels up. Okay, so assisted membrane transport. This is kind of touchy because assisted membrane transport can actually be passive or it can be active. It's the in-between. So assisted membrane transport, they also call it carrier-mediated transport. Things that are carried have two properties about them. I guess there are three properties. Either one is it's hydro. Why can't it slide right easily through the membrane? Why does it have to be held? Because it's hydro. If it's hydrophobic, it can slide through a lipid membrane. It has to be hydrophilic. So things that are that like water. Another thing is something that has a what on it? Positive or negative? Charge, right? I wasn't saying positive or negative. I was giving examples. So loves water, has a charge, or it's just too stinking big. If it's too big, it has to have a special transporter to pull it in. Okay, so there are three important characteristics to these things. These are different characteristics. For it to work, it has to be hydrophilic, it has to be too big, or it has to have a charge. But here are characteristics about those things you want to know. Every transporter is specific. It only works for a specific group of molecules. Like, if it's sensitive to sugar, it only works to carry sugar. In fact, it only works to carry a specific type of sugar. If it's sensitive to sodium, it only works on sodium. It won't carry other things that look like sodium or similar to sodium. It's specific to sodium. If it's saturated, if I have 10 transporters and I have 1,000 molecules, when I fill up all 10 of those, I refer to them as being saturated. What's going to happen to my speed of moving things at that point? It's going to slow down. If you take a sponge and you saturate a sponge, you did what to it? You filled it completely full of water. How much more water can you get into it? No more. These transporters can be overloaded. They can become too full. Like when we talk about the kidney, people that are diabetics, they have too much what in their blood? Sugar. They have too much sugar in their blood. That sugar gets down in the kidney, and your kidneys, your healthy kidneys, can pull most of that sugar back. In fact, it can almost pull all of the sugar back because it has thousands of these little transporters for sugar. But the problem is that if you give it millions of particles of sugar because you have too much sugar in your blood, what's going to happen? Can the kidney pull all those sugars out? Nope. So the sugar goes down into your urine, and then you pee it out later, which gives that sweet urine characteristic. And then competition. Some transporters they can have another molecule that doesn't belong in there, get in there and plug it up, stick to it, and it doesn't work anymore. If you've ever heard of a naloxone clinic or you've heard of antabuse, how many people have heard of either of those drugs? Naloxone blocks opiates, so people that are addicted to opiates like heroin or morphine, they go to a naloxone clinic, and what happens, they give them naloxone and it binds to a naloxone receptor. So, or naloxone, an opiate receptor. Naloxone binds to it. So if they take an opiate, it doesn't work for them anymore. What's it going to do for their pain response? They're going to feel a lot of pain. It's miserable for them because they block all their opiate receptors, and if they get a little hurt, they feel a lot of pain. Alcoholics take something called antabuse. They've got this little enzyme that's sensitive to alcohol, but if you give them antabuse, it's also sensitive to alcohol, and it binds to the alcohol enzyme or receptor, and it blocks it. So if they drink alcohol, they get really, really sick because they can't break it down like normal. Here's a fun fact for you. If you ever have a dog that drinks antifreeze, antifreeze goes to that alcohol enzyme and binds to it, but it never unbinds. So this alcohol enzyme is important for other life functions. If you don't have it, you die. So these dogs drink the antifreeze, it sticks to all of these enzymes, and what happens to the dog? It dies. So if you take your dog to the vet, what's funny is that they look for something that will also bind to that enzyme really fast so that the dog can pee out the antifreeze. So you go there and they open up the cabinet and they pull out their little bottle of Everclear. What's Everclear? It's like 190 proof, isn't it? So it's 95% alcohol. And then they mainline, they give an IV of Everclear right into the dog. How's the dog going to feel? Huh? They would work for a human too. There was actually an episode of House with LL Cool J on it, where LL Cool J was drinking. He was a prisoner and he was wanting to die, so he was drinking toner from a copy machine, which is the same. It's very similar to antifreeze. He was drinking it, and House went in and was getting drunk with him, trying to talk to him and be friends with him. But what was House actually doing? Get that alcohol into his system. They were drinking, I think, tequila, and trying to get it in. So the dog gets smashed off its ass to the point where if it took more alcohol, it could probably die. But what you're trying to do is give it time to pee out the antifreeze and get out of the system. And it's because these little transporters actually are exposed to competition. You can compete for it. And here's just an example. So the last couple of illustrations for today. But here's one transporter. You can see it's like a doorway. Here's your lipid membrane. You've got this revolving door. I always think of P.F. Chang's with that revolving door that spins around and around and around. So you come to the door, you fit into the door, and then it spins you around. Boop, and puts you on the other side. But what about these square things? Are they going to fit in the doorway? Nope. So it's only specific or sensitive to this. How about with competition? Look at the shape. It's round here. This is round all the way around. So this can kind of fit in there, but it wedges in here. Now the doorway can't turn. I just blocked it. It's an example of competition. And then facilitated diffusion is a type of assisted transport. Facilitated diffusion tells you it's what? Active or passive? Diffusion's telling you it's passive. Facilitated diffusion moves things just like passive transport does. It always moves it from what to what? Low to high or high to low?
High to low.
It always moves it high to low. Glucose is a perfect example. In your brain can't just absorb sugar. It has to have a little transporter. And it takes it from the high blood sugar into the low sugar brain. So if your brain is starving for sugar and you're going through hypoglycemia, what's it telling you about the concentration in the blood? It's low. Are those transporters going to pull that sugar into your brain? Nope. So guess what's going to happen to your brain's ability to make energy? It drops. So how do you feel when you're hypoglycemic? You feel tired and weak because your brain's starting to shut down. And if you're hypoglycemic for too long, guess what you'll go into? Right, death. You move right into death. So you get tired, you get sleepy, you start looking like you're drunk because you can't think straight, and then you pass out, you go into a coma. Diabetic coma is who you usually see this with. And then if that's not corrected, then they die. It's because they can't transport the sugar into the brain because they don't have enough sugar in their blood to transport. Active transport is also a type of assisted, but this requires what? Energy. ATP, the energy of the cell. And when we come back, we're going to talk about these transporters like this. Active transporters take something from low concentration to high concentration. So they grab a hold of this particle and they pump it around where it doesn't want to be. You have to have energy to run that pump. So when we come back, we'll talk about something called a sodium-potassium pump that takes what two ions and moves them? Sodium and potassium. It always moves them where they don't want to be, and that's where we're going to pick up at. Yeah, if you have your sign sheet that you read the syllabus and you agree with the syllabus, drop it off because that's my form of attendance. If you didn't bring it in with you, come up and let me know so that I can still count you here. By the way, do you need to do a lecture quiz yet? No, because we didn't finish the lecture. Not until we get to the end of lecture number 1's material. Let me get a piece of paper. Actually, do you have a piece of paper? I'm assuming that you're signing a sheet.